This is the first in a series of podcasts being recorded about the European-funded EiCLaR research project. EiCLaR stands for Enhanced Innovative In Situ Biotechnologies for Contaminated Land Remediation. Today, I'm speaking to Professor Timothy Vogel from the University of Lyon in France, who is the lead coordinator of this project. Podcast number 1 will be giving a general overview of EiCLaR. So thanks, Tim, for giving up your time this morning. To start things off, I'm always fascinated about people's backgrounds, how they get to be where they are, doing their current roles. Just to set the scene, would you be able to give me a little bit of background about yourself?
Yeah, sure, Nicola. It's great to be here, and my background is a little bit unusual in the sense that my French accent is quite weak, as I actually come from California in the U.S. So that's, but I've been in France for 30 years, so that's quite some time. I have actually the dual citizenship, so I can speak French, but we'll do this in English if that's okay with you.
Absolutely.
Okay. And so, yeah, I'm a classic Northern California surfer boy, basketball player kind of upbringing, and in San Francisco. And I was born just north of the city. Basically a West Coast American kind of upbringing, university and everything. It's kind of interesting to find myself in this situation here because actually when I started out my studies in the US, you have a lot of flexibility. So you can change your majors frequently in your undergraduate education. And I ended up with degrees in geology and oceanography, actually.
Wow.
And so that was a bit different. And then I actually worked as an oceanographer researcher after my undergraduate education, which is 4 years in the US, for a couple of years for the US Geologic Survey. So it was quite kind of not in the polluted soil bioremediation area at that time. I don't know whether the dates are important, but it was pre 80s. So anyway,
very experienced.
Yeah, there you go, an experienced person. But you know how things sometimes it's a combination of interest and chance and opportunities that kind of drives you forward, and I think that's really true in my case. I was interested in pollution. And I had been in the US Geologic Survey, been doing work on natural organic processes. And I was kind of like interested in how much that pertained to the pollution that we were putting into the environment. So I went back to graduate school. It's the only place that was within biking distance of where I lived, actually, a university in Palo Alto, California. And I did a master's in environmental engineering. And since I wasn't an engineer, I had to go through all this typical engineering training that undergraduates do that I hadn't done, fluid mechanics and that sort of thing. But so that was different. But anyway, I did my PhD in environmental engineering, but on the biological degradation, the microbial degradation of chlorinated solvents. And at the time, people didn't think that they were biodegradable. In fact, the National Academy of Science in the US came out with a report saying that they were not biodegradable. So it was hard to get financing for my PhD.
So that's the breakdown of contaminants, biodegradable. That's the breaking down of contamination.
Yeah, so these chlorinated solvents have been used as cleaners for degreasing in automobile industry. Dry cleaners used them for cleaning until recently. And so they were quite, people didn't actually think they were particularly toxic. I know I've seen pictures of people with buckets of the stuff washing pieces and things. And so obviously, as science develops, as this environmental studies develop, people found out that these compounds, as it continues to happen, new compounds become fashionable. I wouldn't say fashionable is the right word, maybe, but become a scope of new toxicology knowledge, and then people start worried about them. Then PCBs, PFAS are more recent examples. But at the time, chlorinated solvents were the case. And so I was able to show in the mid 80s that they were broken down, degraded by bacteria that live in the absence of oxygen.
Right.
Anaerobic bacteria. So that was quite novel finding at the time. Obviously, since then, people have developed that knowledge into actual site cleanup remediation technologies. And And so basically from the 83, so for the last 40 years, I've been working on the degradation by microorganisms of pollutants and the use of those in the environment to clean up polluted sites. So
Right.
I guess now I hit 40 years of doing pollution bioremediation, contaminated sites.
Wow.
But so I was a professor at the University of Michigan. I got tenure in the U.S. system. After 6 years, roughly, you get tenure, and then you go on to something else. And at that point, I was looking for a place to do a sabbatical and got contacted by a large multinational company that no longer exists called Rhône-Poulenc in France. And they wondered if I wanted to spend a sabbatical in their place, their research in Lyon area.
Right.
So in 92, my wife's a historian, we moved to France, and I worked for 8 years for the company. And then the company broke up into pieces, and I went back to academic life at the University of Lyon in 2000.
Right.
So, and I went to a department that is actually the Department of Biology. So I'm a professor of biology without actually having a degree specifically in biological fields. So that's kind of interesting. But I do biology. I actually even do molecular ecology, which means I look at the DNA in organisms to understand what they're doing and how active they are, but related to pollution degradation, among other things. So that's kind of, it's a long story to get into France, but that's more or less how I got here. And you, like I say, you never know. I never went back. Came for a year and then another year, and then I quit the University of Michigan and stayed. And now I'm at the University of Lyon. So, yeah.
Brilliant. Well, there you go. Yeah. Everyone weaves a different path, don't they?
Yeah.
So moving on to the EiCLaR project, this is how I met you. Your role, as I explained, is the lead coordinator of the project. So do you want to explain how you embarked on bringing this whole consortium together and a little bit about the EiCLaR project, explaining what it's about?
Yeah, sure. So the EU made some joint agreements with China about 2 areas of doing research in the health sciences environment. And as a result, the EU put together a call for proposals, which would be joint projects with Chinese colleagues. And this started, I think, in 2018, maybe. Time flies. But anyway, and the initial proposal that was done was actually led by some UK researchers and who are actually now on our technical advisory board. And when the Brexit happened, there was the proposal didn't get funded at the time. Brexit happened. So a couple of years later, we decided to go again and hunted for a volunteer for being coordinator of EU projects. For those of you who haven't been involved in EU projects, it's great to be a partner and not so nice to be a coordinator in terms of the cost benefit aspect, but somebody has to be a coordinator. And so I volunteered and basically took off from the proposal that we had done 2 years previously as a basis, looked at how the reviewers were. The call for proposal had been changed too, and they made it much more applied in terms of the technical level that you should reach in the proposal. 2 years before, the level was, I don't know if you're familiar with these TRL levels, these technical, technology readiness levels.
Readiness levels. That's right. Yeah.
And so they go through like 1 through 9 or something, and 3 is roughly the proof of concept.
Right.
And after 6 is mostly in the field kind of demonstration, commercialization. So what the EU wanted was to move things from the lab to the field. And so the original proposal was going like from 3 to 5. And then this one was from 3 to 6. The difference between 5 and 6 is 5 is evaluation and 6 is demonstration. So they want to push it. And so reading the proposal with the partners and everything, the sense was that the industrial side of things was really critical in this call. And so we tried then to identify partners that would help in the promotion, use, application of the technologies that were developed. The EU funds a lot of things, and this clearly was in a program with a mentality that it was supposed to actually develop technologies that would be used. So interest in having French companies benefit from it, but also the environment benefit from it, not just research. And so that was a critical part. And as a result, we had to then start looking for some partners because we had reduction of the number of partners from the UK, actually, as a result of Brexit, I'm sorry to say. But we still have 2 partners in the UK, so it's not like UK is not represented. But the other thing was industrial partners. So we were looking for small to medium-sized companies that would be motivated to use these technologies and find those connections. So we have French, Czech, for example, Finnish companies involved. The other thing that was critical was the communication aspect, and we wanted to have people that were involved not necessarily in the lab or in a company making money, but people were involved either in regulation or oversight or information transfer that kind of was an interface between what's being done technically and the technical information transfer to make sure that people were aware of it. So that was another aspect. And then final thing is that diversity is important. And sometimes people overlook that in projects because you often like to have people who agree with you people tend to select partners who are similar to them, either in background or personality or point of view. And we tried to not do that. It didn't turn out so badly. I don't know. I mean, there's no real serious arguments, but we thought about gender, nationality, kind of job, kind of points of view, and tried to have a diversity in it, which makes the coordinating kind of tough. But anyway, that's a bit of how we went about it.
Okay, no, that's fascinating. And being involved in the project, you do get to meet and learn about people's cultures as well, which is always very interesting. Like you say, people are from diverse backgrounds, not only from research backgrounds, and bringing them from lots of different countries is really beneficial, and I think something of huge value to European projects. So the actual EiCLaR project, it's, as I explained, it's a multifaceted project. Do you want to give a little bit of an explanation about what we're trying to achieve? What's it all about?
Sure. So the bottom line is 4 technologies. We identified in our proposal and in the project 4 technologies that we want to develop and apply. And people should know that bioremediation now has been going on very strongly. The market for bioremediation has been quite strong over the last 30 years. And there are lots of technologies that work which use microorganisms either that already exist in the ground or are added, but harmless for humans, organisms that are added to degrade pollutants. So the area of bioremediation is quite an active area. And that part of the reason it's active is because it's relatively inexpensive. The bacteria work for free. And unlike incineration, where you have to use a lot of energy, or chemicals, where you have to use a lot of chemicals, bioremediation is appealing because, in fact, it's low cost. The problem is that it tends to be slower than these other technologies. You go to a site with an oven and you just burn everything, it's quite quick. The organisms don't necessarily work that fast. And I was mentioning the anaerobic organisms I worked with during my PhD, they're even slower than aerobic, ones that use oxygen. And so there's some organisms that are quite slow, but they're inexpensive. So the concept behind the proposal was to do a merging synergistically of non-biological techniques and biological techniques to enhance the speed and performance of the bioremediation. Because another aspect of bioremediation that sometimes is criticized is the percentage of the pollutant that's degraded is not necessarily 100%. There's when you burn everything to just ash, everything's gone pretty much. But in the microorganisms, the concentration gets down to a certain amount in the environment. There's accessibility. There's lots of issues that lead to the fact that there's always a residual, usually when you use biological technologies. So we were trying to get rid of that residual, if we could, and to enhance the speed of the remediation. Now, getting back to what I said earlier, of course, that might add an extra cost because we're adding physical chemical things which are not free. But so it's a balance. And the other thing that we tried to emphasize in our proposal and in our work is the sustainability aspect of it. So, and the whole aspect of the ecosystem and, what impacts the ecosystem long-term, short-term. We know from other studies sometimes we've done ourselves, that biological treatment has less impact on the ecosystem. So we also don't want to have a large impact, but we want to combine the techniques. So that's the general idea. What happened then is we had 4 specific technologies in mind. One where we use micro and nanoparticles of iron, not dangerous nanoparticles of iron, to degrade pollutants, and use electricity to regenerate the surface of that iron and have microorganisms degrade the metabolites, the products of the reaction, so that everything gets traded. So combined electro, nano, bio remediation. So a combination of all 3. So not just bioremediation. And we were able to base some of that on work that some of our partners had done with electro-nano bioremediation without the, electro-nano remediation without the bio part. So that's one area. Another area that we've been working on is the use of an organism that our colleagues in the Water Institute in Karlsruhe in Germany have discovered that degrades chlorinated solvents, what I mentioned before, but in the presence of oxygen versus in the absence of oxygen. And these organisms are quite interesting because they don't need other substrates, because some aerobic degradation of chlorinated solvents is done, but you have to add methane or ammonia or something. This organism lives on trichloroethylene, for example, as its sole substrate. So you can add the organism and degrade that. So that's one other technology.
So it lives, on that one. So it lives on the actual contaminant.
Right.
And it uses, oh, wow. Okay.
Right. Because today, if you have a chlorinated solvent spills, and every country has many of them, or PCBs, any chlorinated compound, which for those who don't know, we use chlorination of organic compounds to produce really interesting industrial chemicals that were good as solvents or heat stabilizers or things like that. So the industrial world produces a lot of molecules where they artificially put chlorine on organic substrates, and they became large chlorinated molecules. Anyway, the work I had done in the 80s showed you could degrade them in the absence of oxygen by dechlorination, which takes the chlorine off the molecule one after the other. Then people later showed that they could use organisms that degrade methane or ammonia in the presence of oxygen. The enzyme, the way they degraded the methane and ammonia, that enzyme could attack the trichloroethylene, but you had to keep feeding the organism methane or ammonia, and then it wouldn't degrade the chlorinated solvent if it had the methane to eat. It's like the steak and peas thing. You give steak and peas, the person's going to eat the steak before they peas. So you have to feed them the steak so that they can be eating, and then slip the peas in when there's no steak. So that's basically what you have to do with the trichloroethylene. Stop giving them methane so they'll degrade the trichloroethylene, but they can't live on it. Like no kid I know can live on green peas. So so it's the same kind of problem. You can't just give them green peas. You have to kind of go back and forth. So this organism doesn't require that. It just lives on the chlorinated solvent. So that's the second technology. The third is based on the whole kind of field of microbial fuel cells that has been developed over the last 20 years. And the originally microbial fuel cells was to produce electricity from waste degradation, because there are bacteria in the world who, when they degrade a compound, they actually give the electrons outside of their cell. So you can put them on an electrode and they will give the electrons to the electrode, and you can produce electricity. So originally the idea was, wow, a great way to do waste treatment instead of doing waste treatment like we do now, or sewage treatment, that sort of thing. We could use this and produce electricity. The technology hasn't advanced far enough to change our sewage treatment plants to this technology. Although in the lab, we're able to produce electricity, charge telephones, that sort of thing. But it's based on that idea. And what this concept is that in a site that is anoxic, there's no oxygen present, because they have a large, for example, pollution of hydrocarbons, and the organisms have degraded all the oxygen that is in the environment, and so there's no more oxygen, so the hydrocarbons don't degrade very fast at all. You stick an electrode into that environment, but it acts as an electron acceptor. It acts like as a surrogate oxygen in a way, because the other end of that electrode is tied to a system that's in oxygen, presence of oxygen outside of the site. So you stick the electrode in, it acts as an electron acceptor, a surrogate oxygen, and it enhances the degradation. So that's what we're calling bioelectrochemical remediation. But basically, we're using electrode surface to enhance the electron acceptor condition as a surrogate oxygen.
To stimulate it to break down.
Speed up the rate. So it doesn't speed it up a lot, but the anaerobic, under absence of oxygen, degradation of hydrocarbons, for example, is quite slow. Fortunately and unfortunately, because unfortunately for a contaminated site, and fortunately for the reservoirs of oil that we use to fuel cars and everything else, because they are not easily degraded because they're without oxygen. Then the final technology is what we call enhanced phytoremediation, but it's basically combining electrokinetics and plants to degrade a site. So I say enhanced phytoremediation. Some people, when I say phyto, they think of a dog named Fido. Hey, phytoremediation, but it's phyto, and using plants where the roots can stimulate bacteria in the rhizosphere, in the zone where the roots are, to degrade pollutants. And also the plants could uptake sometimes the pollutant, depends on the mechanisms. There's several mechanisms. But one of the problems is the accessibility of the microorganisms and the roots to the pollutants in that soil. And so this technology is using electrodes, pulsing electrical effect, putting pulses of electricity through the system, which is helping increase the accessibility of the pollutant for the plants. So it's electrophytoremediation, if you want. We're calling it enhanced. So our four technologies, is maybe you can understand, there are bio, but mixed with some physical chemical concepts to go further, maybe attack more complex mixtures too, and go faster, hopefully, maybe a little bit more expensive.
But balancing the sustainability aspect as well, making sure that these technologies will be able to demonstrate that you are looking at the social, environmental, and the economic side of these, because that is very important when we're looking at remediation of a site.
Right. And one thing that I have to mention is that because this is an EU-China project, our Chinese partners, who are all academics, there's no industrial partners in China. They have their own funding from China. EU is not funding the Chinese partners, but they interact with the companies, and the consulting environment there is not the same as in Europe. So the universities do lots of what we would call environmental consulting work in Europe for these big companies, and so there's no environmental consultant other than the universities for these companies often. And so our partners there are looking at both the fundamental thing, but also trying these techniques there. And one of the aspects about the sustainability is the evaluation of sustainability the same in Europe as in China? What do we mean by sustainability? What do we mean about when we want something to be ecological? What are the goals? And they're influenced obviously by politics and national laws and regulations. So you see a lot of that effect in dealing with the interaction with our Chinese partners.
Yeah, no, it's a multifaceted project which has got great strengths and challenges. It's a four-year project. We had COVID right in the middle, which was a challenge, or has been a challenge. So the greatest strengths, I would say, with the project is multiple different technologies being looked at, being investigated, being researched. So what do you feel your greatest challenges of the project are?
Okay, yes. So yeah. Well, I think that, as you mentioned, COVID, that threw us a bit, in the sense that with the Chinese partners, as you might have followed the COVID history in China, we haven't been able to meet together until after 2 years of the project. So we had to adapt to that. It was a problem, and we've done a lot of, as everybody in the world has, I guess, a lot of video conferences. But it's hard to do video conferences with people you've never met. It's much easier when you've met them face to face. Then you know how to interpret their interaction on the video. For those who've done a lot of video, and I got emphasis on the video conference. So meeting after 2 years was a problem. because for collaboration, people were getting things done in their own labs, but the interaction wasn't as great as we would have liked it. And that affected also publications. But because the project is really driven by the technologies, it's not such a publication heavy project as some are. I've been in projects where that's our only output is publications, scientific publications. Here we're trying to do the technologies. So there's all the challenges of intellectual property and developing, going down the road and developing the technology, actually making them applied. So that, I think it's kind of a combination of communication and then the practical aspect of actually moving from science, from the lab, from the kind of publication mentality to like know-how or intellectual property application. Who's going to make money off it? Are we going to make money off it? That sort of thing.
And sharing this with the end users, for the end users to then hopefully roll this out in real terms. So that's
right, absolutely. And a good example of our project was to do case studies where I'll say naively, but maybe we weren't naive, but we planned to do that pretty quickly, to contact industrial, not partners in the project, but people who were interested and say, you have a problem, we'd like to do an analysis of what the best technology is for your site, including the typical ones used already, and add ours into the batch. And so it turns out that companies are not so ready, as most people know, to share their information about their sites. So we had to go so far with those discussions to get them to agree just to do the paper study of the case study, that in a lot of those cases, the companies are really interested in us doing pilots on their site. So the disadvantage was we were having trouble getting the secrecy agreements and confidentiality agreements and everything going on it. The advantage was that when we got all that, they were so bought into the interaction that they were saying, maybe you should come and do pilots on our site. So we had a disadvantage that's kind of also in the long term will be an advantage.
Yeah. So that's something that you'll have learned from going forward for other projects. We've almost jumped on the TRL parameters,
Absolutely.
haven't we? Which we weren't expecting to do. So going forward, so what happens to EiCLaR when it finishes? It finishes next year, the end of next year.
So it finishes the end of 2024, right?
Mm-hmm.
EiCLaR ends. The communication part of it, the technologies communication, will be maintained, so that hopefully these technologies will become used already. I mean, before the end of 2024, we're going to be actually doing pilot studies with all the technologies. So that's really good. We're actually doing the demonstration. We're doing commercial evaluation of their potential. The companies involved are motivated to use these technologies. So I would say the real output from EiCLaR will be that these technologies are applied at appropriate sites. So that's I would say the major output. There'll be scientific publications to explain the basis of this and that sort of thing. But I think that for me, optimally, and for Europe, the way they look at this program is that these technologies will be viable and used. And so between now and the end of 2024, we have to have successful pilot studies on actual sites. And they're starting up and they're going to go on, and we'll find out then. So being optimistic, I guess we will say that these 4 technologies will have a life after EiCLaR.
Yeah, and that's the real benefit of this, and having multiple faceted consortium partners. They will look at opportunities to hopefully take some of these technologies forward even further and advance them to other maybe European funded projects or later TRL type projects as well. So
right. So this is the always the kind of the perspective differences between academic researchers and other people. What's the benefit of having an EU project? For an academic researcher, it's not making money. It's discovering new science and doing new technical work and publishing it. And so for those another proposal, another saying, okay, this worked. Can we think of other combinations of technologies that would also work? So obviously innovation continues and they will ask for financing. The companies might also be interested, depends on the company. Some companies might say, this is great. I have the technology I want. I don't need another proposal. I'm very happy. I'm going to go make money with this technology and I don't want to participate in another project. But a company might say, this works so well, sure, I'll add another technology to my group of technologies I have. I'd be happy to add something more. So yeah, I think the reaction, the interaction will depend. I'm hoping that, I guess I'd say, I'm hoping that some people will view it as a real end. Mostly the companies, obviously. I hope researchers keep innovating, but I would hope that some companies would say, oh, this is great, and I don't need to participate in another one. Because some of our companies are fairly small, so they can't actually probably manage 6 different technologies anyway. But the bigger ones, the medium-sized ones, they could be very much interested. We didn't talk too much about all the partners, but the range of partners is quite large because we have groups that are in information transfer, knowledge transfer, communication. We have people that are in business partner, help business develop. So we didn't really get into the diversity of the partners that we have, but we have a lot of diversity, and I'm hoping that EiCLaR will benefit all of them after it ends in some way or the other. Yeah.
Yeah, I was going to say if people want to know more about all the different partners and more about the progress of the project, there is a dedicated website which is EiCLaR.eu. So if this podcast is setting the scene, and we will be doing other podcasts with other partners who are working on the individual work packages. So I'd like to thank you, Tim, for your time and your knowledge sharing about the project and about yourself. And we will upload this podcast very soon. Thank you.
Thank you.